Have you ever wondered how your muscles work when eating your favorite candy? Well, it's your lucky day. We're about to show you exactly how a muscle cell contracts. Let's take a moment to get oriented. A muscle can be broken down into parts that are much smaller than what we can see with the naked eye. Let's take a look at this representation of a muscle, for example. A muscle is made up of many fascicles. A fascicle is a bundle of muscle cells called myofibers. Myofibers are made up of even smaller components called myofibrils. This membrane surrounding the myofiber is called the sarcolemma. The sarcolemma dips between the myofibrils to form T-tubules. The network that you see here adjacent to the T-tubules is called the sarcoplasmic reticulum. The portion of the sarcoplasmic reticulum in contact with the T-tubule is called the terminal cisternae. Let's take a closer look at the components of a myofibril. A myofibril is made up of thin and thick filaments that are arranged in a very specific pattern as seen here. The section seen here that contains both thick and thin filaments is called the A band. This section containing only thin filaments is called the I band. This section in the middle of the A band that contains only thick filaments is called the H band. This zigzag line where the thin filaments attach to one another is called the Z disc. One functional unit of muscle extends from Z disc to Z disc and is called a sarcomere. Now that we are oriented, let's see how a muscle cell contracts. Here is the axon terminal of a motor neuron and a motor end plate. A motor end plate is a convoluted section of the sarcolemma of a myofiber. As an action potential travels down the axon terminal, calcium channels are opened, allowing calcium to enter the motor neuron. This influx of calcium causes synaptic vesicles filled with acetylcholine that are found in the axon terminal to fuse with the membrane and release their contents via exocytosis into the synaptic cleft. The acetylcholine travels across the synaptic cleft and binds with the receptors on the motor end plate, which triggers the opening of sodium-potassium pumps. As sodium is being pumped into the myofiber, the acetylcholine is being broken down in the cleft by an enzyme called acetylcholinesterase. If enough sodium crosses the membrane to cause the inside to reach 55 millivolts, an action potential is generated. This action potential travels down the sarcolemma and into the T-tubules, which causes calsequestrin, a molecule in the terminal cisternae of the sarcoplasmic reticulum that stores calcium to flood the myofibers with calcium. The calcium binds to the troponin complex on the thin filament of the sarcomere, which pulls tropomyosin off the active site of the actin. A myosin head bonded with ADP found on the thick filament then attaches to the active site. The myosin head releases its ADP as it uses energy to pull the thin filament over the thick filament. A muscle attraction has occurred. An ATP molecule now binds to the myosin head, which then releases from the actin. The calcium releases from the troponin complex, allowing tropomyosin to again cover the active site on the actin filament. As the calcium is being pumped back into the terminal cisternae of the sarcoplasmic reticulum, the ATP on the myosin head is being converted to ADP. This causes the myosin head to return to the high energy state, and the whole process can now be repeated.